Hey guys, welcome to our first video on host defense system. This is chapter 12 on innate immunity, and we're going to work through these slides. I have our second set of slides already on Canvas, but these ones, and these ones will be for that first section on innate immunity. Let's get to our PowerPoint. So this will just be kind of our walkthrough, the overview of our defense system altogether. We'll talk through the first and second lines of defense in this set of slides. And as said, I've already pre-recorded the stuff last semester for your other stuff for the inflammatory response, which is part of your second line of defense. And then chapter 13 will be on our third line of defense, which is our adaptive immune system. So before we get into this, there are some major concepts, but this will be basically the rest of the stuff we're talking about for the semester, with the exception of a couple of chapters. So overall, the concept of immunology, this is the study of all features of the body's defense mechanisms. And you may think of this as simply like fighting infectious disease, but actually the immune system is much more complicated and involved in many more processes than simply fighting against pathogens that come into the body. So our immune system is responsible for surveillance. We have immune cells called leukocytes. These are our white blood cells, and they survey everywhere. They're infiltrated in all of our tissue. They go to every surface, so all of our mucosal surfaces are totally coated with protective features from our immune system. We have constant detectors, these things called neutrophils, that circulate in our blood and are looking for any signs of anything. It could be pathogenic infection, but tissue damage due to bruising, right? Even that's part of our immune system response, or a tear that you might get in your tissue. If you have organ disease or some kind of toxicity to one of your organs, then your immune system can detect these things too, right? So it's not just infections, it's really everything, any kind of damage that may be happening to your body. Cancer is the big one too. So when your cells mutate and start looking and producing things that are not normal essentially to your body, your immune system is usually responsible for detecting those things and getting rid of them before they turn into tumors and infiltrate the rest of your body. So surveillance is a big one for the immune system. Certainly recognition of foreign material, and this is definitely where pathogens come in. So if there's a virus or a bacteria or a helminth, a worm, or protist that comes into your body, those are all considered foreign, and your immune system is certainly responsible for recognizing them as foreign and then essentially deciding what to do with them. We have numerous bacteria, viruses that are all a part of our normal flora, as we've talked about throughout the whole semester. So it's important that your body doesn't just get rid of every single foreign entity that comes in, but it is important that it can detect the ones that are harmful and destroy those immediately. And also keep your normal flora in check. I think we talked in the very beginning of the semester, I know I gave you an excerpt from the book called Stiff, and talked about how the body, as soon as it's dead, essentially just gets eaten from the inside out by your normal flora. The guys that are normally super beneficial for you and keep you in good health can totally destroy your flesh once your immune system fails to function. So it's important to keep that in mind, that even the good guys can go bad if not kept in check. Here's an overview of the immune system, and let's just take a minute and talk about this whole concept altogether. So the host defense system is split into two arms, the innate arm and the acquired arm. Innate means essentially you're born with it, right? Like L'Oreal, maybe she's born with it. Okay, so innate means you're born with this. It means that as soon as you come out of your mother's body, then you already have these things in line, and they respond relatively similarly. So this term non-specific means responds similarly, regardless of what comes in, right, to anything. Now I'm going to put a little asterisk because that's not exactly true. We're going to keep it simple and just say non-specific means essentially your innate immune system just says, you're foreign, stop, I'm going to do this thing to you. But I will say that as we get into the details a little bit more, you'll find that that's not 100% true, that there is some specificity to your innate immune system. But when I say that, I just mean like your innate immune system can recognize whether you're working with a bacteria or a virus, whether the bacteria is gram-negative, gram-positive. So there's some groupings that your innate immune system can recognize, but what it doesn't need is to know specifically like which glycoprotein spike is on that coronavirus that's in your respiratory tract right now. It doesn't need to do that. It will respond the same whether you're working with a coronavirus causing the flu versus a coronavirus causing, right, the severe respiratory distress that we're facing today. So that's what this term non-specific means, like a general all-purpose response. For your innate immune system, there are, again, two arms of your innate defense system. The first line of defense is the stuff that you were just built in. So your physical barriers like your skin, your respiratory mucosa, right, your epithelium, which separates the inside from the outside of your body. All of these things are part of our first line of defense. Your chemical defenses are included here, things like lysozyme that's in your saliva and your tears. We've talked about that a little bit already. Your hydrochloric acid in your stomach that destroys almost everything that goes through there, right? And there are actually quite a few innate chemical defense systems that we have built in. Those would all be part of that first line of defense. And if you think of it this way, again, it's kind of this sequential format. So we have these the first barriers that protect us from anything getting in, like our skin. But if you're walking through these beautiful forests behind me and you scratch your arm against a branch, well, your first line has just been breached, right? Unfortunately, we have a second line of defense that is a very, very rapid player. Within seconds or milliseconds, your second line of defense kicks into gear, right? So your second line is activated when there's a breach in your first line, and it does work this way. The second line of defense are your white blood cells that are part of your innate immune system, which means they're sort of these all-purpose responders. So these are our neutrophils. That's the biggest number of our second line of defense, our innate immune system. Our neutrophils are circulating our blood. There's over 60% of all of our white blood cells in kind of a regular steady state are neutrophils. So they're just in high abundance. And I like to think of them like the fire trucks, right? They're the first responders, the first ones on scene in the event of an emergency. And it almost doesn't matter what kind of emergency it is. The neutrophils just get there right away, mostly just because they're in such high abundance and they're really quick to activate. So they circulate your blood, and anywhere you have any kind of damaged tissue, signs of inflammation, signs of an infection, your damaged tissue sends out a chemical alert that says, help me, help me. And then the neutrophils stop and shoot through kind of in a very sequential, very fine-tuned format. They move through your blood vessel lining and then they get into tissue and they can respond right away. But those are not the only cells involved in our innate immune system. Just a second. So there are other cells involved in our second line of defense, our innate immune system, and these would be our granulocytes, like our basophils and eosinophils, etc. So we'll talk about all of these things in these sets of slides. These are also the cells that are involved in the process of inflammation, which is how we're really going to lay the foundation of our understanding of the second line of defense, is by discussing the inflammatory response and looking at each of the cells involved in this process and the chemical communicators that are really key in helping navigate our immune system, helping orchestrate our response. We'll also discuss fever as part of this response. This is not always involved in the inflammatory response, but certainly can provide significant benefits, and we'll talk about that. And then note there is like this branching thing here, gamma delta T cells and natural killer T cells. These guys are officially a part of the innate immune system because they don't have the ability to recognize specific markers on the surface of pathogens. So they kind of have that concept of all-purpose response, but the way that they function is much more similar to the T cells that we have in our acquired defense system, our third line of defense. So we kind of have these as branching cells where they can recognize non-specifically, but their response is much more similar to what we see as part of our lymphocyte response. So we'll mention these ones. I definitely will talk about natural killer cells, and we'll probably talk about gamma delta T cells just a little bit. So over here, essentially when our first line fails, right, we have a second line of defense, our inflammatory response that kicks into gear right away and helps quarantine any pathogen that might be coming in, tries to help reduce the infection or damaged tissue, and contains everything at that site. But sometimes that fails, and that maybe it's a pathogenic infection, maybe that branch had you know, Staphylococcus aureus or something, and it got into your deep tissue and just happened to be that incredibly virulent strain that causes necrotizing fasciitis, and so it really just overruns your inflammatory response and gets immediately into the neighboring tissue and perhaps even into circulation. Now, at that point, our second line of defense is just not sufficient to contain this infection. It's not sufficient to quarantine that thing at that site of injury. And so we have our backup system in place. That's our third line of defense. The third line of defense takes a little while to get going. The third line is called the acquired system, right? The acquired immune response, because you are not born with this one. I mean, arguably, I can talk about that for about a day, but we're just going to say when you're born with this, you really have no significant development of your specific arm of your defense system, which means your body's never seen any pathogens before. So it's never mounted a highly specified, highly specific response. This is called acquired because you gain this over time, specifically over exposure. So the first time you see something, first time you see a pathogen, your body takes about 3 to 5 days to mount a very highly fine-tuned response, right? This is specific, a very, very specialized response. And as mentioned in my example, this would be like identifying the very particular nuances of the glycoprotein on coronavirus and mounting a response that neutralizes that virus by binding to that very, very specific site. And there's really only, this defense line is the only system that we have to do that type of fine-tuned response. Now, the benefit of this highly specified response and the development of this response is that this also provides you long-term immunity to the exact same thing. So one of the questions I've been seeing right now during this coronavirus epidemic is if you can get reinfected with coronavirus or if you can get infected with a mutant version. And my answer has always been it's very, very unlikely to get reinfected with anything, just simply based on the way that our immune system works, that once you've been infected and mounted a sufficient immune response, you acquire immunity to that particular pathogen with a very, very specialized response. And even the mutant forms of the coronavirus, they don't seem to have been mutated enough that our fine-tuned response would not be effective against them. So the idea behind our acquired or adaptive immune system is that once you get this response going, then you have an army that not only can destroy that pathogen immediately as soon as that defense system has developed its response, but a part of your response is called a memory response or amnestic response, and that stays in circulation or in the tissue indefinitely. And sometimes you can lose it over time. And just definitely when you get older, this is called immunosenescence. That's a natural process that your memory immune cells start to deteriorate and die out. But as long as you're healthy, and most of the time, as long as you mount a sufficient immune response, you've developed that memory response that stays in place. And so that anytime you come against that same exact pathogen or even something that's similar to it, your memory cells will recognize it immediately. And instead of taking days or weeks to develop that specific response. You can develop it within minutes or hours of having that pathogen in you again, and so you can eliminate it before it has a chance to set up shop. Now, your third line of defense is where we're talking about our lymphocytes. These are B and T cells, right? So this is lymphocyte. Lymphocyte, these guys are lymphoid cells, so they go hang out in your lymph nodes or in your spleen or other secondary lymphoid organs, and they develop initially in your primary lymphoid organs, which are your bone marrow or your thymus. We'll talk about those in chapter 13, and this is a really cool system, a little bit complicated with how this whole thing works, but hopefully I can break it down for you pretty easily when we get into chapter, excuse me, I'm sorry, not chapter 13, chapter 12. Yeah, chapter 13. Okay, so we're going to start over here, excuse me, with looking at the first line of defense, understanding our physical and chemical barriers, remembering that our microbiota, our normal flora fit in here as well. And then I'll give you an introduction to the second line of defense, essentially talking about the key players involved in our cellular immune response. And then the next set of slides, we'll talk through the inflammatory process and understanding the details behind here. So to understand our first line of defense, this picture I think is quite nice. It shows you a whole bunch of different components or different parts of the body and how they contribute to our initial barriers. Just think anywhere a microbe could enter into our sensitive regions, right? Have access to our cells that they need to get into, or into our blood or into our deep tissue. Any of those access sites, we have protective equipment, physical barriers, and chemical components to help prevent the infiltration of these things into or onto our body. So here's a picture of a bunch of different things that we have in place. Sebaceous glands, which are our sweat glands, right? So our sweat provides a physical deterrent, not just it can help, the moisture can help reduce these guys from getting, or actually like the sweating can help push things out of our skin and sort of run them off of our body, but also the chemical components to our sweat, so our sodium chloride that's in there is very beneficial to our body in preventing any non-halophilic organisms from really sticking to our skin and hanging out there. Our tears have lysozyme in them. And lysozyme is a great chemical defense system. It hydrolyzes the NAG and NAM bonds of the peptidoglycan layer in bacteria. So it really just cuts apart bacteria and makes them really susceptible to osmotic pressure. We have lots of lysozyme in our tears, in our mucus, in our saliva, really any kind of secretion that we have in our body, you'll find lysozyme there. It's a really great enzyme. The tears also, we have a natural like flow to our tears, and that helps push things out of our eyes that gets in there. If you wear contacts, I'll tell you there's a really great image you can look at. It's an electron micrograph of a contact lens after someone just takes it out of their eye upon regular use, and it is massively, massively colonized with bacteria. So if you are a contact lens wearer, then you'll find that there's lots of bacteria there. And so we have these defense systems in place to help prevent those things from causing inflammation or causing infection. Mucus, you'll find lining just about every surface of the body that is exposed to the environment. We have the nasal passage. Our mouth has its own mucins, right? We have salivary mucins as part of our saliva. These are glycoproteins that are really great at just, they're super sticky, right? And so they stick things or adhere things that would otherwise go and bind to our cells, which we really don't want. We have mucin lining all the way down our respiratory tract, down to at least the upper respiratory tract. We don't have mucins down in the lower respiratory because that's where gas exchange occurs. In our esophagus, actually I'm not sure if we have any in our esophagus, but definitely we have in our small and large intestine, we have different types of mucins or mucus that is found lining and coating every surface. There's a huge group of, or not huge, there's a small group of scientists studying this field of mucosal, mucosal immunology is talking about the immune system that's in our mucus and really like in that interplay between the environment and our deep tissue. But mucins themselves, there's a small group of scientists that study mucins, and it's really fantastically phenomenal to look at the beautiful architecture of our glycoproteins, our mucin proteins that kind of fold out from our intestinal cells and give us a really, really needed layer of protection there. In our throat, we have cilia, and I'll show you some pictures of these things in just a second. Cilia, in addition to mucin in our throat, are really beneficial in trapping things and moving them upwards from our lung. So you can see the direction here that these things are really trying to push things up from our lungs. And the direction of saliva and of other things we have is really trying to push things down into our stomach. So if we can get pathogens into our stomach, that's really the best place for them, because we can destroy them pretty readily with our stomach acid, hydrochloric acid, etc. We have bile salts, bile acids that are secreted here in our small intestine that are also really harmful to pathogens too. So let's look at, actually, let me give you just a term here too. One term that I'll use a lot here is called epithelium, or your epithelial cells. Regions are really any part of our bodies, so here in our nasal or respiratory tract, our gastrointestinal tract, even our urogenital tract right down here, and our ears, like all of the regions of our body that are exposed to the air or to the environment have a layer of cells called our epithelium. Obviously that color's not going to work. A layer of cells called the epithelium. Or epithelial layer is the layer of cells separating the outside from the inside. In our small intestine, for example, right? Small intestine, where we have our nutrient absorption, our epithelial layer is really important. Our epithelium actually lays itself down in these villi, and these are lined with cells. And each of our cells in our epithelium has something called microvilli on them, which are important when we're talking about pathogens. Right? These are all cells. There's actually quite a few more of them. Sorry. There's our nuclei inside there. So you'll see these villi in the small intestine and these crypts and valleys, these crypts here, these peaks here that actually have different functions. And in fact, down here we have these really cool cells called Paneth cells that secrete a whole bunch of our defense systems down here in the small intestine. And then if you were to look at the surface, you would find these guys are lined with villi also. So for our small intestine, we have lots of really great defense systems. And then you'll also see lining the small intestine, or I'm sorry, even laying on top of the stuff, we have this mucin, this mucus layer, and it's kind of splotchy in the small intestine. It's much more defined in the large intestine, but this is our mucin. Oh my gosh, I'm sorry, guys. It's 3 of those right there. So our mucus layer, and so these things are really important in defending us against pathogens. In other regions, so this would be the example of epithelium in the small intestine. In, for example, the respiratory tract, we have different types of epithelium, right? These are columnar epithelial cells. And these guys have these things called cilia. And cilia are hair-like projections, and here's our nuclei here. Right, so this is the respiratory tract, upper. Where these cilia are hair-like projections that move things, and they move things that get trapped in the mucus. So in the upper respiratory, there's actually a huge mucus layer. Oops, sorry. A huge mucus layer. It's all mucus, right? This super duper thick thing. So stuff gets trapped in the mucus, and then the cilia move things from the deep lung up towards the back of the throat, where you cough it up and then probably swallow it back down again, right? So these are two different examples of how epithelium looks and their different functions, but really they're important in separating the inside and the outside of the body. So let's look first at the skin. The skin, or our epithelium, that separates the outside environment. I mean, that's the most basic way to think of it from our deep tissue. Has actually quite a few different layers to it. Our epidermis is what you see on the outside. It's thick and it's keratinized, which means it's cemented together with this particular protein called keratin, and it's dead. The outer region is really dead. And it's really cool to watch to really understand how the skin works, and you'll learn more about this in your anatomy class. But we have these really cool stem cells that are located right here, kind of in the basal region of our epidermis, and they grow from the inside, and as they grow or differentiate into our epithelium, then they sort of push the older cells upwards. So you have this constant upward movement of these dead cells as they move up and then they slough off. And these are all really beneficial for preventing pathogenic invasion. So we have bacteria and viruses that land on our skin at all times. And if our skin just was stagnant and stationary, then they could probably get through quite easily. But because we constantly have this regeneration of our skin, where the outer layer is just continuously, daily sloughing off, and it also helps just remove any kind of stuff that's on our skin as well. If you look at our hair follicles, I don't think I have a picture of it. I'm sad that I don't. Our hair follicles, they embed themselves pretty deeply into our dermis. And this has really, really close access to our deep tissue. So usually when we get a skin infection, it's usually in our hair follicles themselves, because it's pretty easy for bacteria, viruses to get, mostly bacteria, to get inside there. But the fact that our hair follicles extrude out, and we've talked about extrusion before, but it's like a slow pulling motion, right? And it really pulls things out with it. So because our hair follicles do that, it actually helps pull out all that plug of stuff, all that bacteria, things that might have accumulated in the roots of our hairs or our hair follicles. They can come out as well. And then of course we have our sweat glands that can flush out the skin and then provide those chemical components too, to help prevent pathogens from colonizing our skin. This is a picture of our stratified epithelium that you see for our skin. And again, you'll get a really deep understanding as to what these things look like more in your anatomy class. But these are the outer layers. There's a whole bunch of different layers in our skin, and these are all dead skin cells on the outside, and more our stem cells on the inside here with our basement membrane. And things move sort of in the outward fashion. So we have this constant regeneration of our skin. And this is as opposed to what you would see, these columnar epithelial cells in our respiratory tract, where we have these ciliated epithelia. I go all the kids to the trampoline. All this ciliated epithelia here in our respiratory tract that serves a completely different function altogether. That mucous membrane that I've mentioned, again, you'll find it in all the different regions of the body that are exposed to the environment. Definitely your digestive tract, really from top to bottom completely. Our urinary tract, really the outward components of our urinary tract, and certainly the upper respiratory tract is completely lined with our mucin or mucous membranes as well. It's super sticky, it's full of glycoproteins. I'll write that term down. You've heard of this before. Hopefully you remember where you've heard of glycoproteins. This is also the main ingredient in the bacterial capsule, right? The glycocalyx layer. So mucus is made of mucin proteins. Which are glycoproteins. And if you recall, glycoproteins means sugar, right? Lots of sugar. And I cannot figure out why it keeps doing that. I know it's just a quick thing that happens when I lift my pen up, but sorry about that. So these glycoproteins, right? They're huge proteins that are just completely decorated with little sugar isoforms all over the place. And this gives them this really sticky adhesive nature to capture things outside the cell. I think I'll try to find a picture for you to put up maybe during our lecture discussion time to show you how cool these unfolding glycoproteins are in the small intestine. They just totally coat and prevent entry of all of our epithelial cells. You find this really everywhere. Blinking in tears, this is not mucus, but this is definitely a part of part of our mucosal membrane, so helping protect our sensitive surfaces. Salivary flow, vomiting, and defecation would be a pretty violent reaction to invading toxins or pathogens, but definitely effective at removing them very quickly. As mentioned, our respiratory tract has these really cool ciliated epithelial cells. This is an actual electron micrograph, scanning electron micrograph of an epithelial surface, where all of these hair-like things, I mean, it almost looks like a coral reef, where you can actually just see these beautiful things just moving with the, I know it sounds really ridiculous, beautiful, talking about the cilia and the mucus, but nonetheless, it's pretty cool to look at the movement of these things. And this is the concept here that with the addition of a gigantic mucosal layer here, the mucus traps pathogens or toxins or food particles or that thing that you accidentally inhaled the other day, pollens or pollutants from the environment, traps them here. And then as soon as they're trapped in the mucus, then the cilia move in a one unidirectional fashion to push things upwards from our trachea up to the back of our throat, where we can either spit them out or swallow them down. This is called the ciliary escalator, ciliary escalator, right, where things just continuously are moved in one fashion from our upper respiratory tract up to the back of our throat, where they can be eliminated. We have other regions of our respiratory tract that are also important in trapping things. Those nasal hairs that everyone thinks are so disgusting are actually quite functional, and I think that they're probably a sign of true respiratory health because they really trap all those things that are coming into your nose, and they can grab them. And the same concept, we do have mucus even when we don't have, you know, an active infection with all that stuff that's building up in there. There's always a layer of mucin that's lining things and helps them prevent things from getting in. The sneezing and coughing are definitely forces of action. If you've ever seen, or if you've never seen a sneeze, I will definitely find this video and post it for you guys on our Canvas page, but picture of someone sneezing is just amazing. These sneezes are so propulsionary to get things outside the body. I bet you could never guess the speed on record, the fastest sneeze. What do you think it is? How fast a sneeze can go? Oh, that's pretty high. Not 200. 120 miles per hour. Fastest sneeze. Guinness Book. I don't even know how you would measure that. Trying to get someone to sneeze really, really hard. Anyway, usually our average sneezes are about 60 miles an hour. 60 miles an hour. We have the force to just get things out of the body. That is really remarkable. And hopefully it helps explain why, even if you're sitting in the back of a classroom, if someone comes into the front of the classroom with the flu or with a horrible cold, then you could still get sick with it. Those sneezes are pretty dynamic in getting things everywhere. Coughs, not as much, but definitely they produce spittle or aerosolized particles. And actually, we can stop here for just a second because this is also pertinent to coronavirus epidemic. The CDC is still trying to figure out what the mode of transmission is for this virus. And if you have friends or work yourself on the front lines in the hospitals, you've heard the constantly changing requirements for personal protective equipment or PPE as the CDC gets new information here and new information there. So initially it was believed to be aerosolized, airborne, right? Which means that the virus itself could just get out into the air. Almost always this means like in a tiny, you know, protective coating, but this is an enveloped virus. So sometimes it can, actually, it's more sensitive to environmental degradation because it's full of lipids. But that was initially thought that it could just get in the air and you could breathe in the particle. And they changed it and said, no, actually, more likely this is, you know, when the virus gets from a sneeze or cough and someone touches the surface, this is fomite contamination surfaces, or, you know, person to person touching someone's hand that just coughed into their hand or something like that. And then they switched to saying, most likely this is aerosolized droplets, right? Which means from sneezing or coughing. And I've talked to you about this. So even just with me talking right now, if you were sitting like my sister is within a few feet of me, then she is breathing in my aerosolized droplets and I hers just from the process of speaking, right? So you generate these things. They're really, really tiny droplets, and they just get into the air. So it's kind of like why they, I mean, definitely why, not kind of why, This is definitely why the 6-foot distancing has been pushed so hard, because the idea is if you can get at least 6 feet away from another person, that those aerosolized droplets that are generated from just regular talking or laughing or sneezing, of course, or coughing, that those particles won't be, they'll be disseminated in the air before they really have a chance to get to you. But if you're in a confined space like an elevator, those things terrify me because of this, and definitely during this outbreak epidemic, if you have a chance to, try not to take an elevator, because that is definitely a confined space that airborne particles or aerosolized droplets can certainly circulate around, even if you're standing 6 feet away on the other side of the elevator, that the airflow would just allow that thing to get back to you. Okay, that was the segue. Let's keep going. Don't forget that part of our primary defense system is our normal flora, right? We have our microorganisms that, if you recall, we called this colonization resistance, that they bind to all of our surfaces, and really what they do is they nestle inside of our mucosal layer. Very few viruses, bacteria actually get down to our epithelium themselves, so that's actually quite inflammatory to us. But they dock on all of our mucosal surfaces, and especially in our colon, we have this outer mucosal layer that's separated from our inner mucosal layer that's separate from our colonocytes. And on the outer layer, you'll just find huge swaths of bacteria. And, you know, as long as you're healthy-ish, then you have beneficial flora that are lining your intestine, and really reduces the opportunity for pathogens to even find a place to bind. And not only that, of course, we've talked about all the other things that provide colonization resistance, competing for nutrients, for example, training the immune defense system to not react to these beneficial guys, but to be reactive to others, and to actually have improved regulation, etc. And then even bacteria themselves have their own metabolites, their own defense system that they secrete out into their environment to kill off other organisms. Fungi do this very well, but there are definitely some bacteria, even not the bad guys, but some good guys that do this too, that help protect against pathogenic infection. Okay. So those were all just primary defense, all physical defense systems that we have in place as part of our primary immune response. But we also have chemical components to every single one of those that I've just described too, right? And to separate these things is a little bit, you know, nuancy, but it is important to understand the difference here. This lovely picture that we're looking at is, I don't know if you've guessed it so far, but this is an ulcer, a stomach ulcer. And the reason I have this on here is because this is it shows you, actually it's a kind of an interesting story here, but our stomach is completely lined with full of hydrochloric acid with a really, really low pH. You know, as we age from infancy to to childhood to young adulthood, and then in our adults, you know, by the time you reach your childhood, really, and certainly as an adult, your pH has dropped down immensely low. So we have a pH of down in regions of pH of 2. When you're a baby, it's pH of 5, 6. Actually, when you're born, it's almost pH of 7. So it takes a little bit of time to get that pH down pretty low. But pH of 2 is incredibly toxic to almost every single organism, and it is effective at killing just about everything that comes through it, except, of course, organisms who are adapted to that environment. And in fact, we do have an organism that is adapted. It's called Helicobacter pylori. Now, the stomach also has a really great protective layer, so it is completely lined with these huge mucins, these glycoproteins, and you can kind of see evidence of that kind of shiny gleam in this picture there. But when you know, when things get out of balance, when our pH is adjusted, or when our stress levels, you know, alter the lining of our stomach, then we can be more susceptible to these ulcers. And for a long time, it wasn't understood like what this actually is, if it's simply just a physiological thing or if it's an infection. In the early 80s, they began to identify that many of these ulcers, and definitely peptic cancer, were associated with one particular organism, which was Helicobacter pylori. And physicians started to, you know, get freaked out a little bit and say, oh, H. pylori is a horrible stomach pathogen, so we should just start treating people. So we should start treating people with antibiotics to clear H. pylori if they test positive for this. So in the 80s and 90s, I suppose, maybe early 90s is when there was a huge push for H. pylori eradication from our patients. So you yourself or your parents may have been treated for this H. pylori in the stomach, and it doesn't cause stomach disease, except that it was associated with ulcers and definitely associated with peptic cancer. So after, you know, this huge eradication campaign, there was a little break in time, and then about 10 years later, there seemed to be an influx of patients with acid reflux disease and leading to esophageal cancer. And when they started testing these patients, they began to see a trend that a huge percentage of these patients with esophageal cancer were treated, you know, 10 years ago to remove H. pylori from their stomach. And so this is a great example of what we call a pathobiont, one that is normally actually quite beneficial. It seems that H. pylori does prevent acid reflux disease and therefore the secondary complications of esophageal cancer. But on occasion, it can become pathogenic in and of itself in the stomach and lead to these ulcerations. Ulcers aren't always caused by H. pylori, but oftentimes they are associated with that. All that, again, another little side story. Okay, so there are lots of different types of chemical defenses that we have. I'm going to list a few more here that I do want you guys to know. So lysozyme, I've already talked about. Lysozyme is a great enzyme. We have this all over the place. Lysozyme attacks the NAG and NAM, so it breaks NAG. If I write slower, maybe it won't do that. NAG and NAM bonds. So it destroys that bond right there between in the peptidoglycan layer of bacteria. So super beneficial, or I would say this is super damaging for gram-positive bacteria. They are very susceptible to osmotic pressure when they lose that peptidoglycan layer. For gram-negative, they have that lipopolysaccharide layer that really prevents them from having an effect of lysozyme. So we have another enzyme called lactoferrin. Lactoferrin is super cool. It has many functions, two broad functions. One, it's really great at sequestering iron. Oh my goodness, I can't even stand that. Let's try that again. One, it's important in sequestering. And what that means is that it essentially acts like a sponge. Pathogens need iron. They need iron for their cofactor in many of their enzymes. So our lactoferrin, and there are a couple other parts of our iron production line essentially that work together like this, but lactoferrin is a key enzyme. It can bind to iron and essentially just kind of absorbs it all from wherever it is, so from our blood or from our mucosa. And by sequestering this iron or absorbing it up, then it reduces iron's availability for pathogens. So it can be really great at reducing pathogens' ability to reproduce. But also, really cool component of lactoferrin, it's really susceptible to proteolysis. So our enzymes in our digestive tract, and for babies, this is also in milk. Actually, it's in high abundance in milk. And we have these enzymes, these proteases that go and cleave off different parts of lactoferrin in a very specific format. And the peptide fragments that are left over actually have the ability to bind to the lipopolysaccharide layer and cause perforations in that LPS layer. So they are antibacterial. So the digested fragment is antimicrobial. Holy cow. And specifically, it perforates the LPS, right? So it makes the outer membrane, LPS, more permeable. sorry. So the cool thing is that lactoferrin and lysozyme usually work together. So lysozyme is great at really destroying those gram-positive cells. For gram-negative, lactoferrin can digest or can perforate that lipopolysaccharide layer, which then allows lysozyme to get in and digest that cell wall that's on the inside, and then makes these guys super sensitive to their environment. So these enzymes are really, really great. There's a whole other set of enzymes called defensins that you should know about. These all have antimicrobial properties to them too. Let's see if I can get rid of as many of these stupid things as possible. There we go. So these defensins are produced by cells in our small intestine. There are other regions of the body that produce them too, but they're really, really important in mucosal defense, and they combat many different types of pathogens and can directly, degrade them too. So many of them have the same sorts of properties that they can bind to that lipopolysaccharide outer membrane. And then there's a whole another group that we just call them peptides, antimicrobial peptides. And again, just as I mentioned up here, that these digested fragments of lactoferrin can be antimicrobial, there are actually quite a few examples of this that are found in like bovine or human milk, that are also found in the small intestine, that many of our proteins during the natural process of digestion form these small peptides that have antimicrobial properties, and most of them are great at doing exactly what we mentioned there, is going and binding to that lipopolysaccharide and perforating it, or making it more sensitive to disruption. So these are some of the key ones that I certainly want you guys to be thinking about and to be aware of: lactoferrin and lysozyme, and defensins, and antimicrobial peptides. There are actually a whole bunch of other ones, but we're not going to talk about them here. We've talked about hydrochloric acid in the stomach. Don't forget about the sodium chloride on the skin. I'll add that one on here too. Salt on the skin. On the skin, right? And then the nasal passage too. That protects us against almost every type of transient organism. They can be there for a minute, but they really can't reproduce there. The intestine has bile. Bile is very important in lipid digestion. It has a bunch of other digestive enzymes too, but certainly key in lipid digestion. If you think about it, lipopolysaccharide, these are lipids, right? So these lipids are also sensitive to digestion from this bile salts here. And then again, antimicrobial molecules like we've just mentioned here. Inherent within every person are these small nucleotide polymorphism, single nucleotide polymorphisms, or SNPs that we say, that give us our inherent differences. This could lead to really, really minor variations, say in like our cell surface receptors. And maybe these receptors are important in like binding sites for pathogens, or binding sites for our immune cell communicators, like our cytokines or our chemokines. So because of this just very low level difference from one person to another, we have diversity within a species as to our inherent genetic susceptibility to cancers, to whatever it might be, right? But then also important to note also here is that genetic variations between you and your cat, for example, are the biggest reasons why you don't get infected with the same pathogen that your cat does, or vice versa, because the viruses, we already know, have really, really fine-tuned glycoprotein spikes that require a really specific type of receptor on the surfaces of our cells. So when you're comparing species to species, even similarly related species, there's usually enough genetic difference between those organisms to provide difference in the physical structure of our proteins that makes those viruses not bind to the same thing. This is a super cool study. I'm going to continue to refer to coronavirus because that's where we are right now, but There's a cool study in Nature that came out just a few weeks ago that was talking about where this thing originated and trying to find its reservoir in nature, and did a really cool genetic analysis of these different coronaviruses in different species, and to see sort of how this thing jumped from one species to another and then kind of morphed its way into the human population. And it's all based on those glycoprotein spikes and the specificity to the same types of receptors that are found in different species. So it's super cool to actually like look at the taxonomy behind, or the evolution behind, pathogens and their host, and to watch how viruses in particular, because they mutate so readily, but bacteria do this too, just a little bit more slowly, but you can see this co-evolution, and also how a mutational event that can happen in a virus makes it really poorly adapted to its current host, but then somehow, you know, able to bind really, really well to a different host altogether. And usually that's when you see a propagation in a different population. Anyway, so that's the genetic susceptibility angle. Just recall that this is a part of our primary or first line of defense, just because it gives us our inherent innate sensitivity or lack of sensitivity to different things. This is an exercise. We'll do it in our lecture discussion. It's really a good opportunity for you to think through everything we just talked about, and see if you can come up with a physical barrier and a chemical barrier that we have innate to each of these different parts of the body, including, like, number 3 would be the trachea, number 2 is the oral cavity, 1 is the nasal cavity, 4 is the eye, 5 is the skin, and 6, you can just say this is like the armpit, because it's slightly different from the skin, but there are different things that we have going on at different sites of the body. But for now, let's just continue this. We're going to talk through these cellular immune components, and usually in the lecture, this is where we would really slow things down and break you guys down into groups, but obviously we're doing this online, so I'll just talk you through these different things with the hopes that you'll work through this stuff on your own, and then during our lecture discussion times, whoops, we'll talk about this stuff too. So recall that what we're talking about here for our second line of defense, that means if those physical barriers that we just talked about are broken in any way. So that would be, for example, let's just go with coronavirus, because that's where we are. Let's say you got coronavirus, someone breathed or someone coughed nearby you as you were passing by them in the store, for all of you who are terrified of that, and you breathed in that particle. And actually this is that's a ridiculous practice, but because I think about this stuff all the time, if I have to go to the store and I'm shopping and I have my 6-foot distance, if I have to pass by someone going in the opposite direction in the store, I'll always breathe out while I'm passing by them, and I do it inherently because I think about this stuff all the time. But if someone happens to be, you know, just kind of coughing a little bit as I'm walking by them, even with that, you know, trying to maintain that 6-foot gap, there's always a chance that you'd be breathing something in. So if I just breathe things out as I go through, then I think that I reduce my chances. But who knows? So this is in the case that you did happen to breathe that thing in. Those viruses happen to have massive virulence factors that are able to really get in, bind to some of our key or ACE2 receptors on our respiratory tract, and get into those cells right away. So they have high virulence. They're able to kind of hijack our systems in a hurry and bypass that primary line of defense that we have in place. Let's say on our other example that you're walking in the woods back here and you cut your arm, and you immediately broke that primary line of defense, right? And so actually that's a really good way to think about what's happening on the cellular level as part of your inflammatory response. Let's just kind of keep in mind you have a breach in your skin, in part because in your textbook, that's the image that it provides for you. So we'll just stick with that. So what happens when your first line is breached? Well, you know, there is a call to arms. There is a trigger sent to your third line of defense to develop your B and T cell responses, but that takes so long. Your body mounts an instantaneously inflammatory response right at that site of injury, whether it's inside your throat where you just got that virus infection, or if it's in your skin. And this helps, again, kind of try to keep things in that isolated region and combat that invasion, infection, damaged tissue, whatever it might be, before it has a chance to get anywhere else in the body. That's what we're talking about here. Before we get into the discussions of the actual mechanisms of the second line of defense, we have to talk about the key players in there. And so that's what the rest of these slides are about. Now we're talking about the key players of your immune system, your cellular components that are in your blood, that are in your tissue. So if you don't know much about blood, right, if you haven't taken your anatomy class yet, or you're not in the clinical setting, your blood has, can be divided into two major components. You have your plasma, which is where all of the, the richest part of your blood is. This is where all of your antibodies are circulating, all of your chemical communicators, your hormones that are responsible for making your whole body work. This is where potentially your nutrients are, your amino acids, right? Anything that needs to get around the blood into different parts of the body that are not cells, they're all in your plasma. In fact, if you were to look at the studies right now of coronavirus, I'm sorry, but it's just in the news right now. Just yesterday, there's a hospital in Maryland that has been given FDA approval to begin working with convalescent plasma. It's convalescent serum, but it's convalescent plasma. It's just taking that plasma component, which has the antibodies in it, and using that to combat this. It's called immunotherapy. The blood cells are where we're going though. So down here at the bottom, you'll see this concentrated, really dark red thing. This is your red blood cells, but also you'll find your white blood cells in this region too. Just a second. Okay. So what are the components of the blood on the cellular level? Well, of course, there's red blood cells and platelets. We will talk about the platelets in the importance of blood clotting and reducing as a component of inflammation, but we'll save that to the next set of slides. But let's focus on these white blood cells that are found. So I just want to give you a couple of vocabulary terms here so you are aware. Where's my, come back over here. First of all, when we're discussing white blood cells, you'll hear this term called leukocytes. So if I can write it without the leukocytes. Anytime you see the term cytes, this means cell. Leukocytes are all white blood cells, right? All white blood cells. That includes the granulocytes and the agranulocytes. This is one way to categorize these guys. You could also differentiate these based on their involvement in the innate immune system, which actually all of these granulocytes listed are involved in the innate immune response. And agranulocytes are involved in both the second and the third lines of defense. In fact, lymphocytes are exclusively involved in the third line. And these other two agranulocytes that are listed, monocytes and dendritic cells, which you'll see over here. So here are our lymphocytes, and here are our monocytes, dendritic cells, and you can even involve macrophages. These guys are really important connectors between the second and the third line. So they link the second and the third. In fact, these, we call them antigen presenting cells. Oops. We'll use that term a lot when we talk about, when we get into our next chapter. And also even when we're talking about the innate immune inflammatory response. But I'll just mention them here. We call them antigen presenting cells, or APCs. And these guys are really great at taking little pieces of whatever's being digested. So they're great at phagocytosis. They take in foreign materials like viruses or bacteria. They digest them and they take little pieces of them called antigens and they present them to our second or third line of defense, our B and T cells or our lymphocytes, right? So these guys are the link between the second and third. They're responsible for arriving and as part of that inflammatory response to any pathogenic invasion or whatever else might be happening. They take little pieces of those pathogens and then they take them physically to where our B and T cells are hanging out. So we'll look at that in a lot more detail as we talk about inflammation, but definitely when we get into our chapter 13 on the adaptive immune system and understanding this in much more detailed information. This term granulocytes versus agranulocytes, this has to do with granules being inside the cells. So if you look at this picture here, you'll see that all of these granulocytes actually have granules inside them. Neutrophils, basophils, eosinophils, and mast cells. Mast cells, most of us are most familiar with, maybe not because of the name of them, but because of what they are involved in. Mast cells are the primary cells involved in allergies, even like hay fever, spring fever, hay fever that many people are experiencing right now, especially in Sacramento and Davis, that we have a flood of histamine in our sinuses. We have massive vasoconstriction, right, and vasodilation, where we have influx of fluids, and we have blah, 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 blah stuff. Well, that's mast cells. And specifically, it's the granules that are inside here, histamine being one of them, one of many of these cellular components that are built into these granules. So as soon as a mast cell reacts, what it does, any of these granulocytes, what they do is they degranulate. That means they release their internal granules, and whatever's inside those granules, they get out into the environment. Neutrophils, basophils, eosinophils, actually all of these guys have particular chemical mediators called cytokines, which we'll talk about in just a little bit too. Cytokines are the actual chemicals that are responsible for communicating a signal from one cell to another. So granulocytes are great at releasing these cytokines, and they go out and bind to different types of cells depending upon what the nature of that cytokine is, and communicate some type of message to the cells in our body. The agranulocytes, as mentioned, are the lymphocytes and these antigen-presenting cells. These APCs, these antigen-presenting cells, also have a great capacity to secrete out very particular types of cytokines. In fact, macrophages are great at this, but we're going to really focus more on their job as presenting antigens, right? So here are antigen-presenting cells. Just to confuse you, monocytes are able to be differentiated into macrophages and dendritic cells, but there are non-monocyte-derived macrophages and dendritic cells. And there are monocytes that don't differentiate into these, so we'll just kind of keep it as this, that monocytes usually do, they're usually the type in circulation, and then when they migrate into tissue to respond to particular inflammatory events, can actually convert themselves into one of these two forms of antigen-presenting cells, both of which do very similar things. And then here are our other types of agranulocytes, the lymphocytes, specifically T and B cells. You can see they are primarily nuclei. And then when they change into what they're supposed to do, they actually kind of change their form altogether and become whatever their nature is. So B cells, they will usually turn into an antibody presenting cell or secreting cell, and T cells can turn themselves into a whole bunch of different types of T cells, including ones that are involved in inflammation, ones that are involved in regulation, and ones that can help out these B cell responses. So we'll look at this in a ton of detail in chapter 13. You'll also see these other types of lymphocyte-like cells is what we call them, these natural killer cells, natural killer T cells, and gamma delta T cells. These are all almost like derivatives of these lymphocytes, but they actually are derived from a different cell line than the lymphocytes are. And they don't have the specific receptors that T and B cells do. But nonetheless, they are agranulocytes, and they're sort of put over here into this lymphocyte-like category. So this picture and the next one, I like them. I put two different versions of the same picture in. I'm sorry for those of you who are listening at home. A little bit loud. Just to help show you where each of these cells are derived from. Now note that all red and white blood cells, so all leukocytes and erythrocytes, are all derived from the exact same hematopoietic stem cell that's in the bone marrow. Bone marrow is the origin of all red and white blood cells. But there's a process called differentiation that occurs, and differentiation occurs when when parts of the cell, excuse me, I'm gonna sneeze. Woo, pardon me, I'm sorry. Differentiation happens when parts of the body communicate a message to the bone marrow by sending out hormones or cytokines, these chemical messengers that are trying to convey some type of information. For example, if you are anemic, right? If your red blood cell supply is becoming depleted because maybe you have a blood leak, or excuse me, Ooh, jeez. Okay. Or your diet is poor in iron, so you're becoming anemic because you just don't have enough red blood cell supply. There are some particular chemicals that are secreted, very specific hormones that are produced in response to this that help initiate the production of red blood cells. Wow. That help initiate the production of red blood cells, and those messages are secreted to essentially that hematopoietic stem cell to say, hey, we need some more red blood cells. And so as that stem cell divides, then its progeny will actually look different from it. So we call this differentiation, where the progeny has different gene expression and they begin to look like something different and function a little bit differently from their parent cell. So in the early stages of differentiation, you'll see that hematopoietic stem cell can differentiate either into a lymphoid stem cell or a non-lymphoid stem cell. In other words, into what will become a B and T cell or what will become any other type of blood cell. So that's that very first step of differentiation. For over on the side where there's non-lymphocytes, then even this will then have one further layer of differentiation into either erythrocytes, erythroblasts which will turn into erythrocytes. So the red blood cell line versus the white blood cell line, and even from there, there's the monocyte line that is derived from there too. So all of these kind of arrows pointing down, we call this differentiation. I'll just write down that word because I keep using it a lot, so you see where that kind of fits in. Differentiation. Differentiation is when a cell, during the process of cell division, produces offspring that have different gene expression, right? And so they are destined to a different function altogether. And so you can see how these cells are sort of related to each other, but also can provide a massively different cell line altogether. These cells up here, these stem cells are essentially your precursor cells, right? So they don't have a function except for to continue to differentiate during the process of cell division. So here you can have your B and T cell lymphocytes. The natural killer cells are derived from that same lymphoid stem cell, but early on differentiate into something totally different. T and B cells are produced from the exact same lymphoblasts, and only upon downstream differentiation do they turn into either B or T cells. Here, your non-lymphocyte stem cells, again, can turn into any of these variations. You can see something like this is mentioned here, colony stimulating factors. Again, these are signals that are sent by the body to trigger the differentiation of these stem cells into something in particular. And usually this is all based on need. So under steady state conditions, I'll give you some percentages here in just a second of how much of each of these different types of white blood cells are produced on any given time. But when there is an active something, so there's damaged tissue, there's a viral infection, there's a bacterial infection, there's an helminth or worm infection, there's a parasitic infection, a fungal infection, allergies, right? So there's all these different categories. The body will say, hey, we need more of type blank, right? We need more eosinophils to combat helminth infections. We need more neutrophils because we have inflammation. We need more T cells or B cells because we're actively fighting an infectious disease, something like this. Then the body's going to essentially focus more on producing more of that type of cell. So this is actually one way that you can use a differential blood cell count or a complete blood panel or CBC to help figure out what's happening to a person's body. The elevation of different types of white blood cells will help focus the diagnostician to find out what's the invading pathogen or what kind of damage is happening to the body by looking at the response. This next slide, I like this a little bit better. It's a little bit bigger, but here at least it gives you a little brief, little brief summary of the function of each of these types of cells. So for example, mast cells, I've already mentioned to you, is highly involved in the allergic symptoms that you get. So use this one, and this one shows you again these monocytes or lymphocytes here and what each of these cell functions are. This is a table from a previous version of your textbook. I like this one because it does include those percentages. So as mentioned, the neutrophils are the highest percentage of all white blood cells that are produced on a steady state condition, almost 60%, with give or take 5% in all your white blood cells. Eosinophils and basophils and mast cells are sort of the lowest numbers there. As far as your mononuclear phagocytes, the ones that are eating cells, the phagocytic cells, you'll have monocytes, macrophages, and dendritic cells, kind of in that order. And then your lymphocytes, there are quite a few of these in circulation, and it also will change definitely upon inflammation. So these questions I'd like for you guys to do, and I'll have you turn these ones in on Canvas. This would be an in-class discussion question, and maybe we'll have some time during our lecture discussion time to go over some of these questions. But you should be able to go through the information that I just provided for you in the last 3 slides. So using that chart and using that picture that shows you, and even the image from your textbook that gives you a super brief summary of each of those functions of the cells, to figure out which of these basic blood cell components, these leukocytes, are involved in each of these arms of defense, in which percentages, how are they similar, how are they different, etc. But definitely take the time, if you're doing this at home, stop the video and see if you can answer these questions. That will help you in our lecture discussion time, because we'll go over this one again, and I'll have you guys submit this one for some points. Just to finish up these slides, cells communicate, as mentioned, through these cell-to-cell communication proteins called cytokines. And in eukaryotic cells, we all, for our multicellular organisms that we are, we have cell surface receptors that bind to very specific ligands or chemicals, right? Particular structures of chemicals. Just like we talked about enzyme-substrate interactions, there's this really, really key interaction between these ligands and receptors. And the cool thing about eukaryotic cells, actually very similar to prokaryotic cells too, but for eukaryotes, we're super sensitive to this. Inside our body, our cells, we have no cell walls, we just have that cell membrane. Embedded in our cell membrane are all these different receptors, and usually they're receptor complexes. So there's like kind of this outer, extracellular component that binds to very specific types of chemicals or chemical signals. And then there's this, the anchoring membrane region, and then there are these receptors that are responsible for engaging the initial receptor and signaling things inside that cell. So one example of this is something called the toll-like receptor. And actually, your book, I haven't changed this yet, I'm sorry, but let me change it to what your book actually uses. And I don't mind which way you learn this one. But we have something called pattern. Whoops, wrong color, sorry. Pattern. Recognition. Receptors. Pattern, recognition, receptors, PRRs. And their job is to recognize very specific types of patterns by their receptors. And patterns, let's go down to this definition here. The patterns that we're talking about usually are these PAMPs or MAMPs is what we usually use nowadays. M-A-M-P is what we use. PAMP is a specific molecular pattern. So PAMP stands for pathogen associated molecular pattern. MAMP is the more common use today. It's microbial. Same thing. Let me get rid of that. Microbial associated molecular pattern. Right, same thing, associated molecular pattern. So this concept of molecular patterns, let's discuss this for a second. This will make sense to you if you think about it. A molecular pattern is a molecule that is repeated over and over and over again to make up a structure of a pathogen, right? Let's think about, for example, how that bacterial cell looks. We know of a couple of things here. Let's draw a gram-negative bacteria envelope. Let's draw this one with a flagella, for example. So the outer membrane of gram-negative bacteria is full of lipopolysaccharide. Let's stop here. Lipopolysaccharide, if you recall, this is a pattern of lipids and proteins, right, and carbohydrates that make up that lipopolysaccharide layer. So this is one example of a molecular pattern. If you look on the next layer, right, our cell membrane, or cell wall, I'm sorry, is peptidoglycan. Now stop here. Again, if you recall the structure of peptidoglycan, these are peptide chains linking NAG and NAM sugars over and over and over again. Another example of a molecular pattern, right? So here's another example. And even the bacterial flagellum, this is made up of flagellin proteins or actin helices, right? Actin helices. And these flagellin proteins are yet another example of a molecular pattern, right? These protein helices just repeated over and over and over again. So here are three examples of molecular patterns. In fact, these are three examples of microbial associated molecular patterns that we have receptors for in particular. And this, when I mentioned to you, the innate immune system's ability to recognize kind of general groups of pathogens, like, for example, bacteria versus virus, gram-negative versus gram-positive, this is what I mean. So these are inherent on all of our innate immune cells, even our epithelial cells that are not a part of our innate immune cellular components that we just looked at, have many of these different pathogen or pattern recognition receptors. In particular, these toll-like receptors. Toll-like receptors are ones that have the ability, we have I want to say 7, I might be wrong. I think we have 9 toll-like receptors that have the ability to recognize very particular types of molecular patterns. And depending upon which of these receptors are engaged or bound to, will determine the type of response that that cell does. You know, so if, for example, the lipopolysaccharide receptor is engaged, this is toll-like receptor number 4, that triggers a series of events that changes the gene expression inside that cell and makes it secrete out very specific types of chemicals that engage our inflammatory response. Versus if the receptors inside the cell, not outside, but inside the cell, these are different types of pattern recognition receptors called NOD receptors, engage viral parts. So if there's a virus invasion and the viral nucleic acid binds to one of our pattern recognition receptors inside the cell, that will trigger a different set of events that will secrete out antiviral proteins instead of inflammatory proteins, right? So depending upon which of these pattern recognition receptors are engaged, will elicit a different type of response in the receiving cell. So the concept here is that once these receptors are bound, the receptor engagement will actually cause a conformational change in that receptor, exactly like we talked about enzymes having conformational changes. And in this case, the change will elicit a downstream signal event that leads to a series of chemical reactions that will change something inside that cell. Almost always it's to change gene expression in that cell. There's kind of a picture of what we're talking about. This is an example of like toll-like receptor number 4, which is the pattern recognition receptor on the outside of a cell. If this engages with its ligand, which is lipopolysaccharide, this engagement will cause a signal through a series of chemical reactions that happens inside this. The signal is called signal transduction. If you're ever curious about this, there are books written about this. There are hundreds of thousands of papers talking about all of these different signal transduction pathways inside the cell. We are not talking about any of them here, but this is kind of the foundation of immunology, is to understand how these receptor engagements actually change that cell. But let's say this is, for example, a macrophage and it engaged this lipopolysaccharide receptor. That signal will be sent to the nucleus and change the regulation of the genes, so change gene expression inside the cell, and cause it to start producing different types of proteins. Most of the time, these are called pro-inflammatory cytokines. that are secreted in response to the engagement of these external receptors, these pattern recognition receptors called toll-like receptors. Here are 4 examples of them. One that we just mentioned, toll-like receptor number 4, is most classically studied because it is the most sensitive essentially and produces the most amount of these pro-inflammatory cytokines. There's one that detects that particular methylation for bacterial nucleotides so that the cell knows that there's a bacteria and not human or other. There's one that detects that peptidoglycan and flagellin. There are nod receptors, others that are inside the cell that are used to detect intracellular pathogens and engage, causing a different type of response. Right, so these are examples of these pattern recognition receptors and how they engage with microbial associated molecular patterns and cause some kind of a change. Now this change can lead to an event called phagocytosis. I don't think I'm going to show that video. Oh no. Oh, okay. I lost my cover slide. Okay, the initiation or the triggering event here can lead to a process called phagocytosis, and we'll talk about this as a part of the inflammatory response. But let's just look at the mechanisms of phagocytosis here first before we end these slides and then look at inflammation. So this is a video showing you a macrophage, one of these antigen presenting cells, also something called a phagocyte. Phagocyte means a cell that eats other things. Well, I don't know if that's what it means, but that's what these guys do. So phagocytes are responsible for surveying tissue for anything foreign, ingesting them, digesting them, and spitting out the debris for other things to come and clean up in our lymphatic system. And we'll talk about these pattern recognition receptors as we look at this, but let's just look at this quick video and then I'll talk through this process here in just a second. So the job of these phagocytes is to survey tissue. They look for anything that shouldn't be there, including microorganisms like viruses, bacteria. They look for injured or dead tissue. This could even be the result of you bumping against that table this morning and that you killed some of your tissue inside of you, causing that bruising. And your phagocytes are responsible for going and cleaning up that dead or necrotic tissue. Particulate matter, this is pollen or pollutants that come in through your diet or through the air, and your phagocytes, if the stuff is not trapped in your mucus, then your phagocytes can find these things also and get rid of them. And so they ingest them, they eliminate them. Some can extract antigens, but I would say not all phagocytes do this, and then they call for help. This is the process here. So let's talk through each of these steps in phagocytosis, and I think that's kind of how we're going to end. So let's walk through from step by step, and just note that everything we talk about here, if you ever take an advanced immunology class, you'll kind of say like, but that's not exactly right what she said back in microbiology. That's because we're keeping things a little bit more simple than how they really go. But it should be enough information for you to get the basic idea as to how these things work. Okay. Step number 1, actually, let's start here. I'm going to go to a whiteboard. I'll just write this out for you myself, and then we'll go back and look at the images here. Okay. Okay. All right. So here we have our phagocytic cell. And the types of phagocytic cells that do this stuff, and let's just focus on the ones that are APCs. So we're talking here about phagocytosis. And working with phagocytes. Types of phagocytes that are really great at this are neutrophils. Those are our early responders, but there are others that come in downstream after an inflammatory event. These would be macrophages. Dendritic cells. You could even say monocytes can be in this category. Let's focus on this being a macrophage, and the short term for macrophage is M with this, what is that, phage, lambda, I don't know. So macrophage. All right, so we're gonna kind of keep it simple here, and let's just put on the surface this toll-like receptor, pattern recognition receptor. Okay, pattern recognition receptor. And you'll see in the next sets of slides, I'm going to start up a abbreviation list, because in immunology, they love their abbreviations. Everything is abbreviated. As you can see, these words get pretty long. So instead of writing out pattern recognition receptor every time, I'll write it PRR. And I'll also say this could be a toll-like receptor, or TLR, toll-like receptor. Remember, these guys are on our innate immune cells, and they have the ability to recognize foreign things. So first, number one, for phagocytes to do anything, they have to get there, right? So they follow a chemotactic gradient, chemotaxis. So we have our epithelial cells, definitely resident macrophages, resident mast cells, so tissue resident, things that are hanging out like right underneath the surface of our skin. They have this really great ability to just immediately release these chemotactic agents. We call them chemokines. So I'll just write that in here. Chemokines, which are chemotactic agents. Are released from injured tissue. And when I say injured tissue, just note this could be anything on the whole gamut of injury. So this could be an actual injury, like you got a cut, right? Or some type of a wound. It could be a bruise. It could be an organ toxicity. It could be, obviously, it could be a pathogen, bacterial, viral, something that got into your or onto your epithelial cells and are triggering this type of response, right? So injured tissue could be anything along that whole line. But let's just say, with our classic example, you got that cut on your skin, and there happens to be like staphylococcus, or let's say a gram-negative guy, because I'm working with a toll-like receptor. Maybe there was a nasty, what's the environmental pathogen, Pseudomonas maybe, that got onto your skin and into your wound, right? I think I like Staph aureus better. Let's just stick with that. All right, so you have your injured tissue secrete out these chemokines that are chemotactic agents. That means, if you remember chemotaxis, that means the movement towards or away from a chemical gradient. In this case, the chemotactic agents create that gradient from the injured tissue, right, and diffuses out through the tissue, and then it will eventually reach the blood. And once that chemotactic agent reaches the blood, then it can seep into the blood, and that's where any of our circulating white blood cells can actually sense and respond. So these macrophages, which would be monocytes out there, or neutrophils, would be floating through our circulation. They would sense that chemotactic agent and immediately arrest their motility through the blood and pass into the tissue, and then move with that gradient to the site of injury. So that's what chemotaxis is. They have to get there somehow. Now, also, bacterial parts can act like these chemotactic agents too. So there are like these ways that bacteria or viruses can themselves have components of them, but we're just going to really focus on chemokines, because that's the most common way that our white blood cells, our phagocytic cells, will arrive at that site of injury. So step 1 is important. These guys have to get there. If there's no way to get there, then there's no response. And in fact, this is the primary reason why people with diabetes gets really prone to necrotic tissue or infection in their extremities, because they have lowered circulation. So there just is no ability for this information to get from our tissue cells out into the blood, and then for our phagocytic cells to get from the blood out to that injured tissue. So this chemotaxis is crucial and a first ingredient to get these guys to that site. Once they arrive, then we have the binding, right? Engagement of receptors. So there must be physical contact. And again, this is kind of one of those places where I'm going to say, I'm sorry if you ever take a future class, because this is not exactly true, but we're going to keep it simple, right? And let's say that this guy has that lipopolysaccharide layer or something, and it engaged this receptor. So we have our microbial associated molecular pattern, like lipopolysaccharide, for example, right? And that molecular pattern engages its particular pattern recognition receptor, like toll-like receptor 4, for example. And so there's a physical interaction here. And this physical interaction will lead to what we call endocytosis. Number 3 would be endocytosis. Endocytosis is the engulfment or invagination of the actual membrane itself. So the membrane folds in to where that receptor is still here, engaged with its ligand, right? Here's that bacteria, still inside there. And it's important to note that this is not the initiation of digestion, this initial endocytosis event, right? This is just in membrane invagination to form something called a phagosome. In this phagosome, these guys are still hanging out, right? There still is this receptor engagement. There's still this bacteria just sort of hanging out here. Now, again, this is important to note because there are many pathogens who have evolved to allow themselves to be internalized by phagocytic cells, and they secrete out chemicals that arrest phagocytosis right here. And so it's brilliant, right? They can get inside. They're in these enclosed internal vesicles, and then they can just stay indefinitely. In fact, Salmonella is a great example of that, this enteric pathogen that allows itself to be taken up by these phagocytic cells, by macrophages. They get inside these phagosomes, and then they secrete out a very particular group of chemicals that causes the arrestment of the actual digestion, so there's no bacterial digestion, and allows them to move these phagosomes close to the centers where nutrients come in so that they can actually access nutrients. So you'll actually see all these little bubbles, these phagosomes inside these phagocytic cells for these pathogen-infected macrophages. But let's just say under normal circumstances, that doesn't happen. This phagosome then fuses, right? So endocytosis, and that is where these guys get into that initial compartment called phagosomes. But note there's no digestion yet. All right, step number 4 is the fusion of lysosomes to phagosomes to form phagolysosomes. Okay, so we have these phagosomes that are all around. These phagosomes have a huge amount of, we have these lysosomes that are all around, lysosomes. These are regularly produced in actually many of our cells, including our macrophages. And these have in them a whole bunch of digestive enzymes. They have low pH in there. They have reactive oxygen species, if you recall. Let's just mention some of these things. Reactive oxygen species, including hydrochloric acid. They actually have nitric oxide in there too. They can have, they definitely have lysozyme and other digestive enzymes, right? Digestive enzymes. So these guys are enclosed in their little membrane vesicles here, and what we call a late stage endosome. These lysosomes will fuse with these phagosomes, forming these phagolysosomes. And this is where digestion occurs, okay? So in here, now we have digestion of that bacterial cell with all of these digestive enzymes all over the place, and the reactive oxygen species, blah, 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 all that stuff that's in there. So this is our digestion factory. So here we have digestion with all those parts that we just mentioned. And then after digestion, there are two fates for all of those different little digested parts there. Exocytosis, but I'll put a little asterisk, that's a horrible asterisk mark. Exocytosis, so neutrophils do this. They just take this whole thing and they just do this membrane fusion where that membrane is bound. Oh shoot, sorry, that didn't work. Never mind. The membrane fuses, and what you have is just the massive release of all of these virus or bacterial parts, right? And so this is just phagocytic debris. Which actually is pus. And this stuff needs to get cleared out of our system with our lymphatic system, and we'll talk about that more in our next chapter. However, I put a little asterisk mark here because our antigen presenting cells, our antigen, I'll write it out again. Or our APCs, will extract antigens, which are pieces of that digested pathogen. And present them on surface receptors and travel to our lymph nodes to present it to our B and T cells. So this is kind of that process there. These guys will actually take a bunch of these antigens and stick them, sorry, that should be an arrow, stick them on their surface receptors. These receptors are called major histocompatibility complex, which we'll talk about in a lot more detail in the next chapter. And they'll just stick all of these different types of antigens that all look a little bit different on their surface receptors. And so now these guys are fully loaded and they have the ability to take these little pieces that they just digested and take them over to our lymph nodes or our secondary lymphoid organs and present them to our waiting B and T cells to try to trigger that third line of defense. Okay, that is phagocytosis. Let's go back and look at the video. Again, you're welcome to pause it at any time, so I'm going to keep moving my way forward. Chemotaxis, recognition and attachment by engaging those receptors, engulfment into that phagosome, that fusion of the phagosome with the lysosome to form that phagolysosome, where you have destruction and digestion, Exocytosis is that last step, or antigen presentation if this is an antigen presenting cell. Here's a picture from one of your versions of the textbook. Again, chemotaxis, these guys have to get there first, or these guys have to get there first and foremost, usually with these chemicals that are secreted by injured tissue. There's the pathogen-associated molecular patterns or microbial-associated molecular patterns that must engage with toll-like receptors or pattern recognition receptors on these phagocytic cells. Here's our engulfment, so the invagination of the membrane to form these internal phagosomes. Here's that late phagosome where those lysosomes fuse here, and they bring in all of these different antimicrobial molecules, right? So here's those antimicrobials. Note that there's the production of hydrogen. There's a hydrogen peroxide. There's hydroxyl ions. There's hydrochloric acid. No, there's, what is this? Hypochlorite, and various things. There's a whole cycle of events that occur that help reduce these guys or destroy them altogether. As well as digestive enzymes, including lysozyme itself and other components. And so this is where digestion occurs in that phagolysosome. Late stages here is the expulsion of the stuff through exocytosis and producing postulation in the form of debris. But otherwise, these guys can grab out antigens and present them on the surface. There's one more picture. This is from your textbook. Again, this is kind of going start to finish. The chemotaxis, they arrive, right? Chemotaxis by the phagocyte to arrive on scene. At that site, they have the attachment. Actually, this is a much better picture. So we have our pattern recognition receptors on the host cell that can bind to and attach to these very particular microbial-associated molecular patterns on the surface of bacterial cells. This then leads to engulfment or endocytosis into that internal phagosome. This will then fuse to lysosomes to make a phagolysosome. Here we have a whole bunch of examples of these different things that are inside that phagolysosome. This is great. So it talks about lysozyme. These are enzymes that can digest DNA and RNA and proteins. So DNases, RNases, proteases, peroxidases, as just mentioned. And here's a bunch of these reactive oxygen species that you'll see generated in there, including a couple that you're familiar with, which is superoxide anion, hydrogen peroxide. There's singlet oxygen and a hydroxyl anion that you can see in there too. So all of these things are great at destroying, digesting that bacteria, breaking up its envelope, its cell membrane, cell wall, etc., all of its protein components, and then generating little tiny pieces organism, which are called antigens, and these are released as debris, or they can be attached to receptors for presentation. These, as mentioned, the primary phagocytes we have are these neutrophils. If you look at this picture, the neutrophils are granulocytes. Macrophages, dendritic cells can also be found in tissue, but usually in the blood. They occur as monocytes, as you see over here. These all will respond or react with these receptor stimuli. C3b receptors are a part of the complement system, which I think I'll talk about on the next set of slides. They can be activated by our third line of defense too, and we'll talk about that in chapter 13. But we are focusing on these pattern recognition receptors, which engage these bacteria and cause this invagination. This is such a cool picture. And these are locations in the body that you might see these in high concentrations, so greatest concentration of phagocytic cells. And this actually makes a lot of sense because these are sort of some of the locations of where our lymphatic system drains to. So you would find these in concentration at the regions of our lymphoid system too. Oh. Okay, this question here, macrophages perform the final job of removing tissue debris and other products of infection. Indicate some of the possible effects when these scavengers cannot successfully complete the work of phagocytosis. I'll let you think upon that, and hopefully we'll be able to talk about that in our lecture discussion time. I think that's it for these slides. We made it all the way through the first half, and I have the second half already up. So hopefully soon I'll get our rest of our chapters done.